Okay, so Sean from Syngenta is going to start with some sort of background context for the session, and then we're going to hand over to Dr. Deborah Cox, who you can see there from Lagan Valley Scientific. And Deborah is then going to go into the specific of a number of those diseases to help us understand the factors that favor their development and how we can identify them and those sort of cultural strategies that we might employ to reduce the likelihood of attack, which is of course the whole point. And then we're going to hand back to Sean, and he's going to run through some of the control measures that are available to us. But we do want to make this session interactive. And so please do ask some questions in that tab, because today is really all about helping you to put, your practical programs and those kind of maybe monitoring measures that I think Deborah's going to talk about in place to prevent your turf from getting any of these diseases that we're going to talk about. So let me introduce our first speaker, who is Sean Lokes from Syngenta, and he comes from a scientific biology kind of research background, but Sean has specialized in turf in recent years. And Sean is the technical manager for Syngenta, and sort of his daily duties involve providing technical support for all Syngenta products used by professional turf customers. His role includes research trials to help develop practical solutions for turf management issues, and focus on helping us all put together best practice integrated programs to achieve optimal results. And Sean also has a focus on biopesticides and good cultural management and biological controls, which are kind of all obviously going to be very much part of our future plans. Okay, so Sean, can I hand over to you for the first part of this webinar?
Yes, thank you very much for the introduction, Henry. I'll take it from here. So yeah, first of all, I'm going to talk about some of the climatic drivers. And as Henry said, this webinar was mainly originally pitched at a sort of stadium environment. The difficulty is we don't have as much climatic data for stadium environments, so I'm going to have a little talk on that later. So there is some stuff that's relevant for sort of training ground type situations and golf type situations as well. But what we're really trying to think about here is those enclosed stadium environments, because they are a really awkward microclimate. It's like what the Incas used to build with their walled gardens. If you wall things in a certain way, you get the same amount of sun as a flat area, but actually you retain a lot more heat, and you can potentially retain a lot more humidity. So it's a very different situation. So first I'm going to talk about the climatic drivers for the different summer diseases we're going to talk about today. And there are obviously lots of different summer diseases, but we've sort of picked on these as the most important ones. And some of you will be facing some of these diseases, and some of you will have never seen them. But what I would recommend is just upskilling and learning more about the ones that you think you could face in the future, because it's much better to have a plan before it comes to your site rather than waiting. So speak to other people who've had experience, speak to other experts in the area if it's something you think is coming to you. So this is all based on average daily temperatures. So these numbers don't actually look very high at all. You think, well, actually, it's already been 20 degrees. We must be near grey leaf spot territory. It's an average daily temperature. So to get an average daily temperature of 20, it can be dead on 20 degrees for every hour for 24 hours, or it could be sort of up to like 30, 32 in the day, and then much, much cooler at night. So it's a whole way of looking at this temperature data set to make it easier. So me and Deborah have got slightly different numbers for our temperature data, and that's partly because ours is all based on averages, because we can do more extrapolation based on that. But we know diseases start at lower temperatures if there's a lot of moisture, and we know that you can go on into really high temperatures. So these are really quick, easy ways to look at it. But I'm not saying if you're below 17 degrees, you're definitely not going to get brown patch. That's not the takeaway here. These are just the sort of places where you want to start watching once you're starting to get those average daily temperatures. So for grey leaf spot, that would be the top temperature one, 20 degree plus, so long consistent periods of warm weather. The warmer it is, the more pressure you're going to get, and long periods of leaf wetness are also going to be important. So if you're in those 30, 36 degree temperatures, but you've got no leaf wetness, then you're going to be a lot safer. Pythium will be around that same kind of temperature range. It does like it warm as well, and you need that humidity and that water on the leaf as well. Brown patch can start a little bit cooler, so average temperatures down to 17. Again, you're going to need to have a wet leaf, and then leaf spot or common leaf spot will go all the way down to 16 to 18. And again, you're going to need a wet leaf for that. So yeah, water is a really, really important management tool. And the difficulty with stadium environments is you need to use water a lot, not just for keeping the grass plant alive, but also for playability in terms of performance management. You've got very high infiltration rates, so sand-based root zones. So obviously that what water you put on is going, so you're needing to replace it more and more. And obviously there's very little airflow. So once you're applying water, it's much harder for that water to evaporate and leave the leaf, so it's staying there for longer. So anything you can do in that space, water management wise, is going to be really, really powerful. So that's something that maybe we could say you can't control. You've got to water a certain amount. We can't get away from that. So the other factor is temperature, which is the one we're going to zone in on now. So this is the temperature data for sort of outside one of the North London clubs. So we've just taken that for a full year. So you can see it's Glenn's done it the American way of doing dates, but I think hopefully we can all understand it's, you know, January, February, March, all the way through the year. And you can see the warmest part of the year is that middle section. And this is a 5-year average data set for daily mean temperature. So you can see it's never getting above 25 degrees, because obviously a 25-degree average daily temperature is really, really high in the day, and then not that cool at night. It's probably going down to like 22, 23 at night to keep that average. So it masks how hot that day must have been. And then if we overlay what we talked about before, so the different diseases and the different temperatures that they're going to be pushing at, obviously, you know, they can be active below this, but it's when they really become more problematic. Then we can see that. So we can see when actually, you know, there's not that many days outside of the car. Let's think of the car park of this North London club. There's not that many days when we're really pushing those temperatures. You know, there's a few times that's happening. So there's threat in June and there's threat in August. So yeah, quite widespread from the leaf spot. But when we're getting into the grey leaf spot, there's not actually that many days. And remember, to get these diseases to push, we're going to need sort of strings of these days coming together. It's not just a single 5-hour period of it. But what kind of is hidden, I guess, is the fact that, like we talked about before, stadium environments are really different. So we did a study of 3 different stadiums, and from that we found that the temperature data was actually on average 2.96 degrees higher inside the stadium than outside. So that's the issue we've got. So we'll look in a moment at what that does to the data set. So yeah, we know it's very different. An important thing is that's not enough data for us. You know, we always want more data. So anyone who's got some good, clean temperature data for their stadium environment, it would be really, really good if you could get in touch with us, whichever channel you want, or send it directly to Glenn Kirby. That would be really, really good, because we're looking to try and build some models in this space, understand what the real gap is between what the weather models think you should be getting and what you're actually getting. So anyone who's got sort of, we're looking at sort of hourly temperature data, not just the max and mins. So we've got a good data set to work with. So please, please, please do consider sending your data if it's something you're collecting. And then this is that same graph, but pushed up. So what we had before, but everything is moved up by 2.96 degrees. And you can see with that adjustment to the daily temperature that we get a big change. So now the threat goes from June all the way into October. And instead of jumping in to grey leaf spot territory, we actually sit there for some significant portions of time. And for brown patch, yeah, we're there or thereabouts pretty much for the whole of that season, and the same for leaf spot as well. So that's why that actually the weather data says we shouldn't be in too much concern, but actually in those stadium environments, it's much more severe than we would expect it to be because of that stadium effect. If we talk about it in a different way now, so now we're talking about the number of days in a year that you would spend above 18 degrees as your average temperature. And so you can see a load of different regions. So the blue line is the South Coast, North London again is the orange, Midlands, North of England, Scotland, and then the average is this dotted line. And that's the scary thing, is why we're talking about climate change more, is from 2010 to 2023, you can see that's the trajectory, that it is rising, and obviously we're expecting that to continue in the future. So whilst in 10, 11, 12 we weren't seeing so much threat, you're not spending very many days above that 18-degree average temperature. Now it's actually an awful lot more than it was. So now we're trending at somewhere like above 20 days a year. So if a disease needs 3 consecutive days, if you have 20 or 30 of them in a year, you're much more likely to get a consecutive string of 3 of them. Whereas if you only have 2 or 3 days in a year, it's unlikely they're going to come consecutively and be damaging. And so this is for the outside stadium temperature data set. And then if we do that same adjustments, we're adding on that same modifier of almost 3 degrees, then we're reaching 100+ days a year in some of these sites. So yeah, that North London Club is going to over 100 days in some years. So that's where it really pushes. And the same trend line, obviously, yeah, we're seeing a lot more threat from the temperature there. We're spending a lot more time above that temperature threshold. So that is kind of framing the debate. Why are we seeing these diseases rear their heads more? Well, this is the slide to tell you why. We're seeing a lot more time spent at those higher temperatures where we're kind of in those diseases happy place than we were historically. And this is another way of visualizing it. So this is how many days you're spending above that 18-degree threshold. So the greener you are, the less days. You can see, yeah, anyone managing a stadium in rural Mid Wales is going to be pretty happy with themselves. But as you can see down this end of the UK, it's a lot more pressure. So this is the outside stadium environment. But you can see no one with those reds. There's nothing too scary there in outside stadium areas. So yeah, hopefully not too much threat of grey leaf spot, which is what we see. I don't think we've had any reports of outside areas with grey leaf spots so far. If we do that same modification, adding those 3 degrees to all the temperature points for the inside stadium areas, then just think about where you are, and that'll give you an idea of how many days you're spending above that 18 degrees. So you're pushing yourself a lot closer to that that average temperature, which is going to deliver more diseases. So yeah, a lot more of the population areas are covered by these reds and oranges now, unfortunately. Yeah, so that's all that slide is to do, is just to try and give an idea of the difference that those 3 degrees make of how many more days you're spending, up to the dark reds are going to be up to 124 days at 18+ degrees. So yeah, in conclusion on the climate stuff, we know that disease is driven by leaf wetness, and we know it's driven by temperature. And we know that in stadiums, we're dealing with a wet leaf for longer periods of time. If you're in an outside area, whether that's a training ground or a golf course, you've always got that sort of natural wind that's going to be moving away the moisture, wicking that away and reducing the leaf wetness time. And we know that temperatures are around 3 degrees warmer in stadium than outside. Like I said, we'd love more data on that so we can draw conclusions. Is that just for the south, or is that for the north as well, or is that across the board? We know that the climate's warming. We saw that in the trending graphs, which is why we're seeing it as more of a threat. And even if you're not seeing it yet, potential is as we move along that trend line, we're going to see more of it in the future. And we're seeing more and longer periods in those climatic conditions, which are suitable for those summer diseases. So we're pushing into their happy place. And yeah, all the conditions for the summer diseases are similar. So we need to be relying on good nutrition, good cultural practices, considering fungicide programs where appropriate, and can we do modeling to improve things? So we need that data set to be able to support that. But I don't think at the moment we see modeling as saying, right now you need to go and apply. At the moment, the modeling is going to be, right now you're in the danger zone, up your level of scouting. So if someone's going out once every 2 days, it's going out every day and scouting more intensely. So it's really framing that, watching, and then you can see the early signs, because the earlier you catch these diseases, and Dr. Deborah is going to talk about that, the earlier you can get in there and do something, the more effectual your management practices are going to be. So that's me to switch over, Henry. That's my first section done.
Thank you, Sean. Okay, so a bit of background there from Sean. And there was just a couple of comments actually whilst you were on. I mean, first of all, yes, we will share the presentation with everyone, and of course, you sort of plea for data. Most of the stadia use data loggers these days, don't they? So, so I'm sure that there is a lot of data there that hopefully can be shared with you. All right. So what I need to do now is I need to reallocate the presenter. So Deborah, I have just made you a presenter, and whilst you are loading up your presentation, I will improperly introduce Deborah to you. So Dr. Deborah Cox is the Managing Director of Lagan Valley Scientific, who are based in Northern Ireland, and Lagan Valley Scientific operates like a turf clinic for routine testing, and also performs small independent research projects to help us all really better understand plant pathogens and also how amenity products work. Day to day, for Deborah, this mostly involves turf decline diagnosis, but the lab also specializes particularly in plant parasitic nematode management. So Deborah's going to go into the specifics of some diseases now and the conditions that favor their development and life cycle, etc. So Deborah, I'm going to hand over to you if that's okay. Yeah, we can see your screen.
Just to check everyone can hear me okay and everyone can
Yeah.
see my screen.
Yeah, you're good.
Great. Good start. Okay. So thank you for that introduction, and also thank you to all of you for your time today, for joining the webinar. Essentially, my job.
Deborah, all of a sudden I can't hear you. Maybe that's me just having gone deaf.
No, can you hear me now?
Yes, I can hear you now.
Okay, hopefully that's sorted. So I don't know how much of that got through, but just to retrack, my job here today is really to help you to understand some of the main drivers behind 4 plant pathogens that are common in turf, to help you understand what you see, the parts of the life cycle that you see, because you will not see all of them, to help you understand how to be proactive against outbreaks of disease, and to coin a phrase from Glenn and Henry, to help you think a little bit further ahead. So essentially this is going to cover 4 diseases, so brown patch Pythium blight, leaf spot and grey leaf spot, emphasizing the main differences between those 2. Okay, so moving on. First of all, if we think about brown patch Rhizoctonia, this may present to you as blighted turf, and as the name might suggest, it may present to you with a subtle brown or tan tinge. Now, severe outbreaks, to the best of my knowledge, are fairly rare in the UK environment, but as Sean has already highlighted to you, our climate is changing, and so the way this disease will progress, it really likes higher temperatures, and so this is one of the reasons why it's on our agenda. Essentially, what you may see if you stand a little bit further back is the appearance of more rounded symptoms, so circular patterns rather than a diffuse spread of symptoms across the surface of the sward. This is one of the main characteristics if you stand a little bit further back, but the idea would be, of course, to try and identify this before severe symptoms start to develop across the sward. But typically anything which stresses out the turf will really help accentuate the symptoms that you see on the surface. And importantly as well, this fungus will produce mycelium. So this is something that you're going to hear time and time again throughout this presentation, but just as a reminder initially, it will produce mycelium. Now, mycelium, for those of you who don't know what that is, you can think of it as like very thin strand-like structures that's produced by the fungus to colonize and occupy new space. They're small microscopic filaments essentially. And the only stage of the life cycle which you're really going to see is highlighted in the circle at the top left. Essentially, it looks like a fluffy white mass. So this is what we mean by when we say mycelium, a fluffy white mass. Now, the way that this fungus will spread is it will essentially go over the surface of the leaf and create what's called a penetration peg. So you can think of this as a little quite literally like a little peg. It produces a lot of pressure, and so it physically bursts through the cell wall to gain entry into the tissues. They colonize inside, spread around, and this creates this sort of floppy look to the leaves, which turn brown through the infection. The fungus is capable of creating what we call sclerotia, or these sort of really tough structures, which is really just a dense ball of mycelium, which you can see on the bottom left-hand side of the screen. So these will survive some fairly harsh conditions. And so this is one of the reasons why your thatch management is really important, because these structures can overwinter and survive a long time. And they too can be the source of mycelium. So it's not just the spread physically from one infected source to another, it can already be present in the environment. So conditions that really drive this disease, you will see in the textbooks, annoyingly, I understand, a fairly wide range of temperatures, but it's always going to operate on some sort of a gradient or some sort of a scale. I realize that it's frustrating not to have a solid number, but we always operate within a range, because there may be additional stressors that may mean that at lower temperatures you see outbreaks. So additional stresses is always something to consider. Each of you will have very different scenarios, different pitches, different stadiums, different environments. And so it's quite reasonable to understand that if you were to create or to generate your own data, you will have differences between different sites, although you may be physically close together on a map. And so I've highlighted in bold a few things that I think are really important. Number one, I've already mentioned it, and that is your thatch management. So any way in which we can reduce the initial inoculum, because if the inoculum starts off lower, you go through your renovation, you will obviously remove a fair amount of any potential inoculum during that process. But when it comes to, say, overseeding and things like that, those young seedlings, they can be succumb to Rhizoctonia infection. But the disease really needs high humidity for prolonged periods of time. As Sean has already mentioned, these are two really key drivers, both the temperature and the prolonged periods of leaf wetness. And if you look in the textbooks, you'll see that it's typically over 10 hours of leaf wetness is really required. So trying to keep a dry leaf is really one of the main ways in which we combat this disease developing any further. I don't think it's talked about enough, and that is the importance of your cultivar selections. There's many excellent varieties available to you, diploid in particular, that do have really good tolerance against the development of brown patch. And I think a lot of the data that we have really originates from the States as well. So just another thing to highlight to you. Again, everyone's lens is a little bit different, and my bias is obviously towards nematology testing. It's probably the most common thing that I do. I wouldn't receive a lot of samples with heavy Rhizoctonia infections, but they most certainly do come, I would say, more as a secondary or on weakened turf. And so a few things, if you do and find yourself in the unfortunate circumstances that you have an outbreak, things like just changing your mowing pattern to make sure you mow that area last so that you don't transfer active mycelium from an affected area to an unaffected area is probably one of the best things that you can do immediately, as well as improving anything like your airflow or reducing the periods of leaf wetness on the surface of the sward. Pythium, again, this is it's a type of so it's an oomycete, so it's not actually a fungus. So it's a different class of organism completely, and because of this, the chemistries that we use to control fungi are different to oomycetes, and so we have to be a little bit careful when it comes to correct identification of pythiums. Now, it may present to you like some of the pictures that you see now. So one of the key things that happens is the turf will really collapse. And so it can look a little bit greasy. And in the textbooks, you may see this written down as like grease spot. And it really comes from this sort of oily, water-soaked appearance on the surface of the sward. And on the right-hand side, you'll see that under the right conditions, so high temperature and very high humidity for prolonged periods of time, over 14 hours, you will see some fluffy white mycelium form on the surface of the sward on the periphery of where the infection is. And so similar to Rhizoctonia, this organism can also be involved in damping off. So basically during the death of young seedlings, there's a multitude of pathogens which can be involved in that process, but this is also one. So we're talking about the same causal organism, but it can affect plants in different ways depending upon the life stage that they are at. The life cycle of Pythium is quite complicated, and for most of you, from a visual standpoint, the part of the life cycle that you're going to see is the one which is circled for you just now on the right-hand side. This is the mycelium, which will develop under high humidity and high temperatures. But under the microscope in the laboratory, what we're really looking for is the oospores and zoospores, some sporangia, and some of the features in the bottom left-hand corner. But you will really only see those with high-powered microscopy. So I've just included that for completeness. But from your perspective, the development of mycelium will be something that you will see with the naked eye. You don't need a hand lens to see that. It will be fairly abrupt if this disease is actually active. So it's just to highlight that that's the only part of this complex disease cycle that you will actually see. Now, conditions driving this, you can see this again where water is in abundance, particularly where you have poor drainage or any drainage issues, because the moisture gets held and retained in the soil. And what that means is that this organism, because it's so water loving, it really thrives in those environments, and so the zoospores can spread very, very easily under those conditions. And so this is why you can sometimes see the patterns follow drainage lines, because that's where the water flows. So if you have an outbreak in one area, sometimes they can form channels and stripes and lines and things like that. But really, we're talking about very high humidity, over 90%, for prolonged periods of time, for 14 hours, and very high temperatures as well. So again, we have this common theme, which would really coincide with summer, which also really coincides with the time of year when you've got your renovations. And so this could be a challenge for you too. One of the top things on my list, and you'll see in the textbooks quite a large range of things that you can put into place, but measuring your soil water content, I think, is one of the key things to keep an eye on, because the outbreaks of Pythium, they're really driven by moisture. And so making sure that you're in an optimal range for you is just so, so important. Overwatering is one of the key drivers of this disease. So keeping an eye on that is absolutely pivotal, and drainage management is also key, because as I've already mentioned, if you have any moisture retention within the sward, it basically increases the risk. It doesn't guarantee that this is going to develop, but it's just an aggravating factor. Again, mowing infected areas last is probably one of the best things that you can do, because you don't want to be spreading any of the mycelium that could potentially infect uninfected turf. You also don't want to be spreading oospores through additional water applications, if you can avoid it, of course. But certainly improving your airflow and making sure that the period of leaf wetness is reduced as far as reasonably practical are some of the key tips that will help get you through if you do happen to have an outbreak, before you could consider reaching for pesticides. Now, in the laboratory, it's quite difficult to show you some of the distinct features of oomycetes, because sometimes they take quite a while to develop. But most certainly the mycelium, you can look at that under the microscope, and the pictures on the far right hand side are examples of what you might see. Now, to the untrained eye, they might all look very similar, but Pythiums don't have septation, and so these are just the walls between the tubes that you see. So if hopefully you can see my mouse, if I just draw my mouse over this this hypha here, you can see that there's no lines crossing the hypha. This is what I mean by the septa, but here you can see that the arrow is pointing just next to a 90-degree angle that's typical for rhizoctonia with the septa here. So this is one of the key diagnostic features if you have a microscope, to basically look and see some of these key features. But you do need a microscope for this. It's not something which you will see with a hand lens. Damping off, I've already mentioned it, but it's really the reason why it happens. Well, there's multiple, but primarily it's more a condition where the environment is more conducive to the growth of a pathogen rather than the growth of the plant, or you have slow plant growth or poor seed. Maybe the seed is too shallow or too deep. These are all very common problems when it comes to damping off, but it's not just Pythium and it's not just Rhizoctonia. You can have Fusarium as well. You can also have Microdochium taking advantage of a weak plant. So if it's not growing optimally, the risk of damping off becomes high. So this is where things like biostimulants can really come in handy to try and improve the evenness and the speed of germination, to try and get you that little bit further ahead, so you have a stronger plant so that it can cope, because there will always be some residual pressure from plant pathogens. They will always be present. It's just a case of learning to live with them and essentially reducing the risk. I have highlighted here that nematode pressure can play a role in this too, because depending exactly on where you are in the life cycle of pathogens like root knot nematodes, they can become fairly active in the summer months. And so if you have a young seedling which is infected, this will essentially increase stress. And we know that increased stress can increase the susceptibility of particularly young plants. So it's something just to bear in mind. But essentially high humidity, again, a very similar story, and prolonged leaf wetness are two key drivers, but as well as that, dryness as well. So it's a little bit like Goldilocks, just getting the environment to a state which is least conducive to the development of these diseases. Now, you may know this better as Drechsla or Drexla, or whatever way you would like to pronounce it, but perenniphora is what I now understand to be the more common name for a generic leaf spot. Now, lots of different fungi can cause leaf spots, so this is not something which is unique just to this genus, but just to describe to you a little bit about what you may see, because some of the spots develop lesion-like symptoms that could be mistaken for grey leaf spot, and they are very different pathogens which are active under very different conditions. So essentially, from what I have seen, this would typically develop a little bit later in the year. So if you unfortunately do succumb to grey leaf spot and you manage that effectively, but the plants may still be stressed, you can have secondary infection. You can actually have mixtures of this with grey leaf spot, and that does become quite challenging to identify. But typically you would see this later in the year. You'd see it under periods of higher sward moisture levels, I would say, and also in periods where you have low light. So really it's a disease which is more associated with stress than anything else. So it's just a couple of things to be aware of, that this is actually what we consider to be a weaker pathogen, well, when we compare it relatively, of course, to grey leaf spot, which is very different, and I'll talk about that in a few slides' time. On the far right-hand side of this slide, hopefully you can again see my little mouse, but you'll see here there's a little black brown spot with a yellow halo, and it's just to highlight to you that that is something which is very, very common to see. And for example, if I receive a sample into the laboratory and maybe it's spent a little bit too long in transit, you will find these leaf spots. So they are very, very common, but again, the conditions during extended transit time are going to be cool and wet and humid. So these are really the conditions that help drive these spots. So with a hand lens, you may see symptoms similar to what you see on this slide. So there is a lesion-like discoloration in the middle of a new grass blade. So this is an active infection, but it isn't grey. And this is where colour becomes really important to understand, that this spot is actually brown and black. Maybe the light doesn't show that in the best way, but the main thing to look out for with this disease is the coloration, in addition to whether or not the leaf tips have started to become deformed and twisted. You'll see here in this leaf where there's active infection, you don't have the twisting of the leaves. It's actually one of the main key things I would suggest to you to learn about and to look for, because it's fairly distinctive and somewhat unique to grey leaf spot. Actually, you don't tend to see this with other diseases. So the examples where I've basically chopped off the leaf tips of the leaves which have these lesion-like discolorations and spots, you can see that they're actually a little bit beat up and torn, but they're not twisted and thin. So it's one distinction that you can make between the leaf spots which are just due to stress and that which is a fairly destructive and aggressive pathogen. Grey leaf spot, however, is definitely the one to look out for. This, by contrast, can really easily be mistaken for drought stress. It can usually display itself to you as a wide and diffuse pattern, so not in a circular fashion like brown patch would be. And one of the key giveaways that I've already talked about is just this leaf twisting that you see, and I'm going to show you some photographs of that. But just to highlight that the growing stages are particularly vulnerable, and across Europe, to the best of my knowledge, the disease activity really starts between May and June. Now, that doesn't necessarily mean to say that infection starts then. As we know, disease progression takes time. So it could start earlier. So it's just something to be aware of, because the time frame during which you're going to have your growing and the young vulnerable stage of the plant really coincides with peak activity that we would start to see across Europe. We don't know how this pathogen came to be in the UK. It's still a little bit of a mystery, but just a very brief overview. It was initially found by Dr. Kate Entwistle at the Turf Disease Centre in 2020. And since that time, I think we've identified in at least 5 stadiums that I'm aware of, but it could be more. I don't know the full extent of it. But the idea is to keep tracking this, because it could potentially be a problem depending upon the climates of our summers as they unfold. So I would invite you to read, there's actually 2 articles, but the one that's linked here is on the GMA's website, and it actually goes into some detail about this pathogen. It was written by myself, Kate Entwistle, and Sabine, who's based in Germany, and we've really been working quite closely together since that time to try and better understand the spread of this disease and how we can best manage it and combat it. So some of the pictures that I show you here are from that article, and it's just to highlight a few things I mentioned. So the leaf twisting here, which you can see on the left-hand side, this is very distinctive, and you'll start to see that the discoloration in the leaf is more like a lime green. The lime green with grey spots with a hint of lilac. Now, I know I'm being a bit specific here, but colour is actually very, very important. These are not black spots like what you see with generic leaf spot, which is associated with stress. It does look quite different. You also have along the side margins of the leaves, it's almost like a concave. It's almost like the lesions kind of slope inwards on the outside of the plants. So the formation of the spores across there, you might see some of them with a hand lens, but you may struggle. If you incubate a piece of turf, you may start to see what will look like grey velvet across the surface of the leaves. And this example from Kate is really textbook quality, that this is really what you're looking for after incubation. Now, you may not see as extreme as this, but it's just to highlight to you again, we have these sort of indentations, a lime green color, a soft gray with maybe a hint of purpley lilac in there. That's really what you're looking for. But one thing that I must really highlight to you as well, sometimes you can see a blackening or a discoloration towards the crown of the plants, and it's really important not to mistake that for anthracnose. Just because there's two main mistakes that you can make with the management of this disease, and one, I've already mentioned it, is to mistake it for drought. So if you think it is drought, but you don't go out and actually measure what the water content is and put down water, that creates extra humidity, and that will really drive disease. It's been a painful lesson learned across Europe. And the second painful lesson that's been learned is the application of nitrogen, thinking that the turf maybe will recover with a bit of feed. The lush growth, it really is very, very susceptible to infection, but also for the spread of spores. So exponential sporulation can really be expected if you do that. So just something to be really aware of, to really make sure that you do apply the correct treatment, but also just to make sure that you don't do either of those two things if you happen to have grey leaf spot. The life cycle, again, you're really only going to see maybe one part of it, and that's the sporulation and lesions up in the top left-hand corner. But just to draw your attention to the speed at which this can develop, it's very fast. So people have painstakingly sat at microscopes for very long periods of time and monitored how quickly this a spore can land on a leaf, produce a penetration peg, get into the leaf and propagate inside, and it's typically just over a day. And the exponential rise in the number of spores and the opportunity for spread is just so abundant. Is one of the reasons why it's so feared amongst turfgrass growers, because of the rate at which it can spread under the right conditions, of course. So with that being said, what we really find is that on your pitches, while I've talked about moisture being a key driver, the initial outbreaks have actually been in the areas where you've maybe seen more abiotic stress, because the turf is under more stress. It seems to be that the symptoms present there first. So you can do a little bit of an audit to try and figure out where it might be more appropriate for you to do some scouting work. But essentially, this disease really does need high humidity and high temperatures. Now, I've put down here again, sorry, it's annoying, wide temperature range, but it is true. Infections may occur at lower temperatures. We certainly know that this disease, it can originate from the roots. It doesn't have to originate from a spore. And this might partially help explain why we can see recurrence in the same rough place. So as well as maybe being a hotspot for abiotic stress, there may be residual inoculum in the form of decayed leaf litter, which has stayed within the root zone, and mycelium has originated from that material to infect through the root systems. So we know that, but we also know that the prolonged leaf wetness is a key driver. So overwatering or basically control of your water in the sward is really, really important, because the time that it takes for the sporulation to really develop is highly dependent upon it. So the temperature tied to the leaf wetness is really quite key. So if you have higher temperatures, you need less hours of leaf wetness. And this is really one of the main challenges in managing this disease. Again, looking to practical tips that will help you prepare. If you haven't done so already, looking at the genetics available to you, looking at your seed mixes and cultivars. There's some excellent breeding programs out there. I know that Sabine was working on this many, many years ago when she first identified grey leaf spot in Europe, maybe in 2015 or 2016. So it has been ongoing for quite some time. And the pictures that you see on the right-hand side, there's no fungicide applied in these trials, but essentially what will happen is there'll be a natural inoculum and people will sow different varieties to see basically how they fare in a high humid environment under high pressure for this disease to develop. And you can see quite clearly here that there's a fair range of cultivars with excellent resistance. Now, it's not going to be 100%, but there's certainly some cultivars that you probably wouldn't want to include because of their susceptibility. But it's also important to think about any overseeding, or if you do happen to have an outbreak, what seed are you going to use to fill in the gaps? It's definitely something that is worth looking into now if you haven't done so already. If you unfortunately do succumb to an infection, pretty much everything you do is important from that point on. I've already mentioned about watering and I've already mentioned about nitrogen. So just trying to be proactive in monitoring is really going to pay its worth, I feel, because if you can pick up infection early and limit the spread, and then understand that if the conditions are conducive, you may have an outbreak situation, you can work with that. So you have that knowledge, but the presumption always has to be that periods of leaf or extended periods of leaf moisture is really going to be the key driver here. And I've put in at the end of here just about hygiene and just about visitors and walking across the pitch. Now, I have seen some fairly scary pictures of footprints just from people walking across an infected area to an unaffected area. It really can spread very, very easily. So just a note of caution that if you do have an outbreak, be a bit careful, both with what you do on the pitch, but also sharing machinery between a stadium environment and your training grounds. So while it might be less probable that you're going to have a serious outbreak in a training ground scenario because it's more open, so you don't have this enclosed microenvironment that's perfect for the development of this disease, it's always possible to transfer it. And if you have any I suppose airflow issues, or if it's exceptionally humid, you could potentially have disease activity outside as well. So just something to think about in terms of limiting the spread. Now on to so-called like DIY guide. One of the best things I think you can do to be proactive, and this idea does not come from me, Dr. Kate Entwistle has been pioneering this for years, and so I really want to reiterate her message, and that is to try and take some cores periodically, especially during the summer, especially during these high periods of stress, because it may help you be proactive. And if you need a second opinion, you already have something there, because time and time again, we often receive samples after maybe some period of time, or maybe after a fungicide has been applied, and so it slows the development of what is active on the turf. And so we really want to think very carefully about how we can really help each other to understand what has gone wrong. And so the picture that you see here is essentially just a core which is being taken, put inside a grip seal bag, wrapped up fairly tightly and taped in position. Now, this core was actually submitted to me about a week and a half ago, and so it's just to illustrate to you that if you package like this, this is absolutely ideal material to submit to any laboratory, and they will really thank you, because what you don't want is for the soil and the sand to mix with the foliage. It really slows down identification and it can mask the symptoms. So this is really something which you can do. And if you do take a core like this, just something to bear in mind, it may not be so clear from this picture, but there's actually a little bit of an air gap here. So you want the leaves to be in high humidity, but not saturated. Okay, the saturation can actually limit the formation of the spores to help with identification. So humid, but not saturated. So it's very important. Now we do have to talk about textbook worthy symptoms versus what you get in reality. They are quite different. On the picture on the left, this is actually some initial symptoms of grey leaf spot developing. This was after incubation in one of those little grip seal bags like what I just showed you. And hopefully what's apparent to you, if you look closely, hopefully you can see my mouse, but you see here, the tops of some of these leaf blades, they're actually starting to turn grey and lime green from the tip down with a little bit of that twisting going on. You can also see some twisting going on here as well. So sometimes at the start of disease development, when you want to identify it, sometimes these symptoms are very, very subtle. Again, here you can see there's some twisting going on. There's some lime green development here. There's some twisting here. So the symptoms are really quite subtle. If you continue to incubate it and keep it in one warm, humid place, you may start to see the sporulation. This is why just being proactive and taking some cores periodically, letting them sit and quite literally watch the turf grow can really, it can be quite instrumental in helping you understand disease development. But more often than not, this is the kind of symptom which you will see. It's twisted leaf tips, a hooked appearance, a lime green colour, lesions which are grey to violet coloured, and sometimes indentations on the sides of the leaves. That's more often than not what I see. And if you don't have perfect sporulation like this, maybe with a high-powered hand lens, you might see something like that, but you really need a microscope in order to confirm this through the spores. They have a fairly unique shape. So it's just to give you an idea of the kinds of things which you will see at the early stages of infection. So we can ask ourselves just a few questions, almost like a tick box or like, I suppose, a checklist. You can think to yourself, well, what does my surface symptoms actually look like? Is it a diffuse pattern? Does it look a little bit like drought, or is it more circular with maybe hints of brown? If at that point you're like, that doesn't look quite right, let's take a core and let it sit. If it develops mycelium, grey leaf spot doesn't do that. So you can rule out certain things straight away. But if you do go down this route, you can also ask yourselves things like, well, what's my water content like? Does the drought symptom actually make sense compared to the data that I have? Is your air light and is your airflow and is your light sufficient? I've put that in purely from a standpoint that I would typically see more stress-related leaf spots later in the year. But certainly in the middle of summer, maybe light wouldn't be such an issue. But these are important questions just to ask yourself. Is there anything here that could be going wrong? So you could go and check your data, check your logs, do a little bit of investigation, maybe a little bit of an audit to try and understand if there are any risks or if there's anything going wrong, because as I've already mentioned, two key mistakes is over-application of water and over-application of nitrogen driving lush growth, and that can really drive disease. If you do have a diffuse pattern, maybe it's maybe a more unusual pattern, but you produce mycelium, again, at that stage, grey leaf spot doesn't do that. But if you do start to see these lesions and leaf twists in particular, it's maybe not entirely conclusive of grey leaf spot, but it's leading you to a better conclusion, to be better informed. And at that moment, if you do want a second opinion, the picture that I showed you where it's actually tightly wrapped, you can open that up like a tin can. You can literally take a pair of scissors and go around, lift it up, get a pair of tweezers and pull out some leaves and actually look at them. If you're not sure, just fold it back over, put it inside a new grip seal bag, tape it back up and send it to a lab. That's all you'd have to do. You don't have to go through any complicated like stitching and putting things back together or opening it up. It's really quite straightforward, and that would really help any lab to help you essentially. So just to very briefly touch on at the end of this talk, a couple of things which are being worked on in the background for your information. There's some spore trap data, or spore trap samples, I should more accurately say. I've been trying to work with some colleagues in the south of England to try and understand if we could repurpose some samples which have already been taken over the last maybe 4 to 5 years. These were originally collected to look at things like pollen and pathogens of trees. Now, because of this, the sort of location of these spore traps, the data could actually be repurposed to try and screen and see, could we actually detect the spores to better understand the risk coming from spores, because these spores can lift very high into the air. We don't actually know how it came to be in the UK, but one entry point could be through wind. So I'm interested to learn if there's anything in the historical data that might suggest that we could actually maybe pick this up early to inform us better of risk. You can detect through qPCR in the thatch, in the roots, and in the leaves before you start to see lesions. That is possible, but it is like looking for a needle in a haystack. So if you remember that picture that I showed you of the core, which is all wrapped up, I don't know if any of you have ever seen like an Eppendorf tube or a DNA extraction tube, but they're like maybe the size of my pinky. Okay, so you have a big core, you still have to have the skill to go through that, select the right material to get it into some, like maybe, I don't know, the size of my fingernail. You know, you're really looking for a needle in a haystack, so it's not perfect. So this is why having some documentation, some photographs, some idea of where, if you have had historical issues, go back to the same spot. We have the technology now to map that really accurately. So you could go back to the same place and you stand a better chance of picking it up to know whether or not you have a risk. Again, before the symptoms develop, it's ideal to know, but sometimes it's a case of confirmation, and by the time you have sporulation, by the time you see the lesions, you can more readily identify that the spore. There's also some interesting papers coming out looking to identify this grey leaf spot through like a lateral flow device. So it works a little bit like, you know, like a pregnancy test or one of those COVID lateral flows, which I'm sure you're all familiar with. I think that would actually be ideal for the field, because then it's a test which literally takes minutes to complete. At the moment, there's prototypes for the strains which cause disease in wheat. It doesn't use the same genetic marker that would really be infecting ryegrass, so it's not applicable to us right now, but I think it's a step in the right direction. So I'm quite excited to see how that would develop, because I think that could be a really useful tool for rapid diagnosis in the field. So just to touch on some final points, your challenges and solutions are going to be unique to you. There is no blanket sort of one temperature or one, I suppose, water content value that's going to serve absolutely everybody. No one knows your turf better than you. So keeping an eye on things, monitoring, collecting data, and keeping photographs, as painful as they may be. I usually say the uglier the better. They will serve you down the line. Future you will thank current you for taking them. Share them with scientists. I know that the guys at Syngenta are always keen to work with you and to try and develop models. I think that's a good step forward. So please do reach out. We should always be prioritizing our cultural practice. So I've highlighted many things, but the reiterated message for me is really water management, trying to reduce stress in any way that we can, and essentially looking at what cultivars may potentially serve you best, because some of these newer varieties that are coming out do show very good promise indeed. So something definitely to think about. And so with that, I think I will pause here and ask Henry to come back on.
Deborah, that was brilliant. Really, really brilliant. Thank you so much. But oh my gosh, there is so much to unpack there, isn't there? So look, I've made some notes. I can't understand any of them because, you know, there's so you raised so many kind of points. But I mean, I think, you know, just to sort of have a bit of a recap at the end, what I'll do is I'll allow Sean to load up again. But, you know, obviously, you know, especially in stadia environments, you know, we've sort of got the sort of perfect storm going on, haven't we? We've got those higher temperatures, we've got that humidity, we've got that leaf wetness, airflow issues. It's kind of the perfect environment for sort of these diseases which you've talked about, in particular, to take hold. Okay, and so, I think one of the key messages that you're sort of coming out with is actually, it's not a black and white situation, is it? Is you have to understand your own environment. You know, this is not about someone telling you or what to do, or it coming from a textbook. This is all sort of, you know, as turf professionals, we need to understand our own situation, our own factors, and how things are changing all the time, and it might be leaning us into different situations. But, you know, essentially, we've got a problem with the way in which we're trying to re-establish pitches in stadia environments. You know, the conditions aren't conducive to it, like a proper integrated strategy. It's not like we can sort of, you know, sort of, okay, we'll avoid lush growth or we'll avoid leaf wetness and stuff like that. Those are part of the challenges you were saying.
It's a big ask, isn't it? It's
you know, it's really tough. It's why there's so many people on the call. Now, firstly, I think sort of, you know, with potentially there being new diseases like grey leaf spot that we're not familiar with, identification is the issue. And I think that was really helpful when you were talking, especially with grey leaf spot, about the colour being distinct from sort of, you know, normal leaf spot, but that twisting and that sort of fish hooking of the leaf being, you know, really characteristic, and no mycelium as well. That was the key part of grey leaf spot, wasn't it?
Those are the two things I would say.
That's really, really helpful. And of course, that bag test that you were saying, you know, that Kate's been sort of advocating for a while now, can be helpful. But, you know, also it's sort of, you can keep your eye on it, but then you can also send it away to yourself or Kate or whoever it is if you feel like something's happening and you don't know what it is.
Absolutely.
Yeah, so we can sort of, you know, we can sort of do sort of ident, and then you sort of talk, because you talked about, which I think is a really good expression, you know, be proactive in monitoring, you know, and that is kind of, it's not to sort of like get caught out after the event or sort of, you know, or sort of be reactive. It's to be proactive, and so you sort of mentioned, I think that sort of pre-symptom testing is starting to be explored, isn't it? Have you got any sort of I know you sort of said it's a bit of a needle in a haystack thing, but have you got any kind of further thoughts on that?
No one is going to know the stadia better than the ground staff that manage it.
Okay.
100%.
Well, that's important, isn't it? Because what we don't want to do is it's about empowerment, isn't it? Rather than sort of evolving to someone else and getting someone else to come up with the answers for you. This is all about people understanding the management of their own environmental conditions, but also being all over and having quite a systematic, proactive approach to monitoring.
Yeah, I mean, performing some kind of, even a small audit just to say, well, where have you seen symptoms before? Can you really narrow it down? Because in that case, you could maybe monitor that area. It means that you're not searching absolutely everywhere. You've got some sort of an anchor or a marker that you can use. And that maybe may help newer staff to try and identify it early too, because you might not always have eyes on this yourself. So sharing knowledge and information is just so important.
Yeah. I really like that, the whole collaboration kind of,
Yeah.
you know, probably going to be a big part of us kind of successfully managing this is we need to understand from each other as well as understand our own situation. And so sort of, you know, people sharing their experiences and sort of talking to each other and sort of getting, probably taking an industry approach would be really important for these new things. Now, how do you feel that grey leaf spot's going to go? You know, you sort of said we don't know where it's come from. There's 5 sites where it's been positively identified. Are we sort of like, you know, just in this kind of, you know, the period before the storm where it's almost like it wasn't about, but now it is about, and so and once it's in, you know, do we expect this to becoming like an escalating problem because we know it's here now?
I mean, I certainly don't want to be responsible for fear mongering,
No.
but I think that it is a very important point that what we have seen is establishment in the US, gradual progression across Europe, tinkering in the south of England, and then every year since it was first identified. Now, that this is of course perhaps biased because maybe not everyone submits a sample. I only see a fraction of what really goes on, but my understanding, and it's been incrementally found over time.
Yeah.
And so as our climate is changing, I think it's just important to understand the risk. It is a real risk.
Yeah.
Reaching out to European counterparts, I think, is really important because they would have already gone through the trials and tribulations and may be able to offer you some advice. So networking is really important.
Yeah. Okay. So just because we, you know, yeah, I mean, I don't want to fearmonger either, but it's almost like it's, you know, what we're sort of probably should be saying is kind of like, you know, let's be proactive with our monitoring because there is a chance that, you know, that it is potentially an escalating situation. And it's quite a serious situation because of the speed at which grey leaf spot develops, for instance, that let's just be kind of aware that it's about and kind of like and set our sort of, you know, proactive monitoring in place, and let's be talking about it to people. Let's kind of like just make sure that we're aware that it's a potential issue rather than, you know, start panicking about the whole thing. Okay, now you did mention grass types, and there's been a question on that, which you may or may not. I know you're not sort of a grass type sort of expert, so but I'll ask it anyway.
Okay.
In terms of grey leaf spot, have any of the tetraploids shown resistance to grey leaf spot? I know we've got the diploids. We have our own grey leaf spot sort of variety mixed around that we kind of recommend in those kind of high risk situations. Do you know anything more about that?
Yeah, you're absolutely right. I am not a grass breeder or an expert in that area. However, to the best of my knowledge, there are, what it really comes down to is the actual individual varieties and their individual genetics. We can't really put a broad blanket over every tetraploid. I mean, tetraploids will typically be a bigger plant. They'll have greater coverage. You know, there's a lot of unique traits about them, but some of them can be very susceptible. So it really comes down to the breeders doing trials and then sharing that trial work so that you understand the risk of the whole seed mix, because obviously it's going to be a mixture with certain percentages of different varieties in there. So there's a wide variety available, but the best people to reach out to are the breeders themselves.
Yeah. Okay. Fair enough. Absolutely brilliant, Deborah. Really, really fantastic. Really thoroughly interesting and engaging, but just really helpful as well. And so, look, stick around because there may be some more questions, and you may want to just sort of chip in.
Yeah. But, don't be shy, people.
We're going to move over to Sean. So, have I made Sean the presenter? Oh no, I didn't do it. Sorry, Sean. I got halfway there, Sean.
You said all the right things.
So what we're going to do is we're going to sort of move on to control measures, aren't we, Sean, with the sort of technologies that are available. And this is particularly important because Deborah touched on this in the sense that Pythium is a very different organism to a fungal pathogen, and so the sort of identification and sort of knowing what it is that you're trying to control does make a massive difference in terms of what you would reach to. And so you're gonna very much touch on that, go through each of the diseases, and then you know, give everyone the heads up on what they should be using, but also what they shouldn't be using.
Yeah, that's right, Henry, and that's why we've laid it out in this way. You know, first of all, we talked about the specifics of climate, because that frames what disease you're going to be potentially looking at and or not looking at. Then Deborah's gone into the details of IDing, and really that does feed into what we're talking about now, because if you can get a good ID and if you know what you're dealing with, then you can be a lot more prescriptive about what your treatments are. And that really is the crux of this presentation, is choosing the right product, applying it in the right way, and that's going to have a huge impact on performance. So if you have a product that's not right for the disease you're targeting, you're not going to get good performance out of it, and you're going to be complaining to me, and that's going to be understandable. But it's about education and making sure we've got the right products for the right pathogens. Different fungicides have different ways of working. So generally, if we're looking on a scale of warmer weather to colder weather, systemics are going to be much better in the warmer weather. The reason for that is they're going to need a level of growth. So if the plant is not moving, if the plant is not growing, and I know for most stadiums, that's pretty much not relevant. If you have underfloor heating and if you've got grow lighting, there's always going to be an element. So I wouldn't be scared about applying a fungicide in January if you've got underfloor, if you've got undersoil heating and you've got grow lights on there. You're going to be keeping that temperature up. So wanting to use systemics when it's warmer, wanting to use contacts when it's cooler. So Heritage would be something that very much fits in that. It's a systemic acting product, so it needs to move through the plant. If it's got a zero growth, then it can't move through the plant to the areas it needs to impact. It's going to be the same for Acernity. You guys have had it in stadium environments for a while. We got it in for golf situations last year. Last year. So that's a double systemic, so 2 systemic mode of action AIs in there. So again, it's going to need that growth to be able to move through the plant. If it's absolutely freezing and there's no grass growth, then you're not going to get it pushing through the plant to the areas we need it. FL321, it's got a bit of both, as well as in Strata Elite, so those kind of can go in those in-between areas. And Medallion's going to do a really good job still, even if it's really cold. So it will still be a useful product in those other times to sort of go between different products, but it's going to be really powerful in cool seasons as well. So first on to the leaf spot. We'll talk about that one first. And I guess to start off with, it's kind of highlighting a bit what Deborah said, that leaf spot is probably, of all the ones we're going to talk about, the least concerning of the diseases, in that it's a fairly weak pathogen. So we do see fairly good efficacy with products when we apply. So if we can get good control early on, we generally see good results at controlling leaf spot. It's just a bit of a weaker pathogen that's kind of targeting grass. It's a bit more stressed, or it's undergoing some other pressures from somewhere else. So in this trial, which was done at the STRI in Bingley, these are the different treatments we had. So untreated, and then a lot of different fungicide options. Preventative applications are going to be highlighted in green. So it's 2 applications for each product, and curative applications are going to be in orange. So first, in terms of the untreated control, so this black bar shows the untreated, and we're just looking at percentage disease here. So obviously, the higher up the graph we are, the higher area of the turf was covered by disease. And generally, if we put a mean of all of the different preventative applications we got, this is what happens. So by preventative, these green arrows are when they went down. So it's going down early, at that time they only just started to see disease, very first sign of disease, or slightly before first sign of disease, and then again a little while after that, and that's when you're doing those applications. And we see good levels of control. We're able to keep that leaf spot nice and low. So yeah, you can see all the products performed really, really well. FL321, you can see there's the purple line, so that looks to be performing the best in that situation. But as you can see, there's no statistical differences between those. So all those products that we tested performed really, really well against the leaf spots. So the important message there is just making sure you alternate that chemistry. If you have a favorite product or combination, just making sure you do alternate that, so you're not potentially breeding for any resistance in the future. And then a very similar trial, so this one curatively. So instead of applying before we get really bad disease, it's kind of targeting it and once you've got disease in place and it's raised up a little bit, that level. And the only difference here is that the two applications was Acernity and then Medallion, or then Acernity, then Heritage, then Acernity, then High-Gear. So again, you can see the untreated, the black line, and then this is the trend of the overall curatives. So yes, it's already hitting sort of 4.5% disease coverage, which is fairly severe. And then that's when we're going to the first application and then second application, sort of late May. And you can see, again, similar situation. The FL321 is the blue this time, performing really well. And then the other products all there or thereabouts. So yeah, all doing a good job for us in terms of disease control. But we would say if it was just leaf spot and you'd identified that right and you were happy that's what you're targeting, then FL321 would probably be the first choice, but then making sure you rotate that chemistry. And that's the overall picture. It's what we always say, if you've got the option of preventative or curative, always going preventatively gives you the better data. You know, you've applied the same amount of product, you've spent the same amount, but you're getting better results preventatively. And I know that's a really challenging thing to do, because how do you know you've got disease until you see that you've got disease? But it's really, the earlier you can get that scouting, you know, the further down this line you can be, the better your results are going to be, the lower you're going to be able to hold it. But again, the products do a really good job curatively as well, so don't be afraid to use them if it's already got out of hand. So to conclude with leaf spot, as Deborah mentioned, you know, resistant cultivars is something to explore. If this is something you're seeing year on year, ask the question of your seed provider, what is it like on leaf spot? We're having problems. Do we need to look at a different variety or cultivar? Anything you can do to control organic matter for the reasons we've talked about. Good management practices like sharp blades. Again, moisture always key, so allowing the leaf blades to dry out. Anything you can do to increase airflow is always going to be beneficial. And make sure you do a fungicide management plan. So look at what happened last year. You know, were you happy with the results you got? If you have leaf spot at the same time every year, just start to think about that and say, right, well, every year we apply it this time. Maybe if we went that 1 week earlier, are we going to get better results? And just play around with your program and build that into a plan, record what you do, and then review it at the end of the year. I know nobody has time to do these things, but if you can get really, really good results of just looking at what went right and what wasn't as good as what you would like it to be. And Syngenta leaf spot models. So we've got a Grace leaf spot model. We're looking at that. We can build a leaf spot model if it's something you guys want to feedback, if it's something that you are having pressure from and you'd like more models on. So moving on to grey leaf spot now. So where is the challenge? So yeah, generally anywhere it's warm, but we aren't seeing it move into more northern climatic areas. Yeah, as time goes on, Dr. Deborah's touched on these already, but the main drivers are those warm to hot temperatures. No airflow is going to really push it because it's going to mean that leaf remains wet for longer. High nutrition and young grass plant situations, so grow-ins especially could be a challenging time. So Glenn commissioned a study with Dr. Deborah actually at Lagan Valley in 2022. So this was not with a strain from the UK, but a strain from a German football pitch. So could be a potentially different strain to what we've got in the UK, but hopefully it's indicative of what we've got because it's coming from Europe. And the study is in a Petri dish, so like the Petri dish we can see here, and it's essentially just looking at mycelial growth. So it's a really early stage study to say, this is what the mycelia would do if it's left unchecked, to just grow over the agar. And then if we applied a product, how does that check the growth? Obviously, the less growth there is, the better control the product is offering is the assumption. So it's looking at resistance testing, but also screening for AIs for their efficacy against the disease. So products that are in our portfolio and those outside of it. The big takeaways were, Acernity and Instrata Elite both performed really well in the study. We've got 2 fungicides coming in the pipeline that will hopefully be here in the next year or 2, which both looked very promising. And there's a biological in the pipeline as well, which was looking good, which was nice to see. But there may be some resistance issues in some strains of grey leaf spot to azoxystrobin. So that's the active ingredient that's going to be in Heritage, but obviously it's available in a number of different products as well. So that was a watch out. So we'll talk about that a bit more as well. So yeah, these are the different products which were involved in the trial. Obviously, we've had to redact all the bits with the sort of ones that are in our early pipeline stuff. But you can see all of our products were included in the trial. And these are the results. And overall, it's really good news for chemistry. So this is in terms of percentage control. You can see the bottom there, the Instrato Elite, the bottom green line, giving really, really good levels of control in line with the Cernity, and those orange ones are those new to pipeline products we've got coming, so they look really promising as well. So yeah, it's good to have some options there. Medallion looking like it's doing a pretty good job there in that in vitro study, so 80% plus. And biological one not performing so well. And then this is the watch out, is that heritage there with a negative value, so that means it's growing faster than in the water control. So this might be that it's unsure as the reason, but we want to flag it as a watch out, that we want to be careful in that space, and we're going to do more digging into why that is. And that might be that it isn't the case for the UK strain, but it's something we've seen in this German strain. So yeah, it's something we want to be proactive about. So yeah, in terms of our traffic light party, grey leaf spot lab study, we would say Cernity and the Strata Elite would be our go-to options. You get your lab results back, you've got grey leaf spot, or you do that ID test where you're seeing the sort of black mauvey grey fungus on the leaf. Then if you know it's grey leaf spot, then going for Cernity and Strata Elite as the first choices. Medallion as a good intonation partner, so building that into the program, but not as your first choice necessarily. And heritage at this time, we would say, as a watch out until we have more information on what our strains are like. So yeah, moving away from heritage for grey leaf spot. But also, yeah, there's two new bits of chemistry looking good, and biologicals could also have potential as well. So the last one was a lab study. This is moving on to a pot study that's done in the US. And I wish I had more data to present to you guys, but part of the issue is because grey leaf spot is only in those stadium environments, it's very hard for us to do trials. We do trials in outside trial stations, which are not seeing severe disease pressure. So if we set something up at the STRI for grey leaf spot, we can get them to put a coloche over the top, but we're not guaranteed to get pressure. So it's really hard for us to run those studies. So that's why the studies you're seeing from us at the moment are at these early phases. But this is a journey, and we're continuing to invest to see what's going on in this space. So yeah, this is a USA pot trial. And you see a very similar situation in this bigger scale. So the reason you do a pot trial is you just get there's more information. There's a living plant involved in this, which makes it very different to what's going on just in a petri dish. And then again, it's never going to be as good as what's going on in full plot trials or full field trials, but this is an intermediate step. So this is disease pressure. So the higher you are up, the worse the disease pressure is, the higher the percentage coverage of the disease. So yeah, untreated, really, really high disease pressure by the end. And then down the other end, Cernity, Strata Elite, and the two new products that we've got coming, looking really, really positive in that space. So doing a really good job in the pot trials. And then one of the biologicals looking really positive there as well. But Medallion not performing as well as it did in the other one. But again, every test is going to be slightly different. We would still say it's an okay as a recommended intermediary partner. And biological 2 looking like it does something, but again, not very much compared to the untreated. So again, we would take the same outcome from that. So heritage would be a watch out, Medallion as an intonation partner, but your go-to choice definitely would be Cernity or Instrata Elite. Just as a watch out, I know some people look on the US website for recommendations. This is the system they have for all the different products and how they rate them against the different diseases. For grey leaf spot, heritage is scored as a 3. This would be back when they did their original data set on it, so this could be 10 or 20 years ago, so it might be that resistance has developed since then, so they're scoring as a 3. So we would just say that as a watch out to, if you're going on looking at different data sets, just make sure it's relevant to your siting as well. So we would say to discount that for the moment. And yeah, the really important thing is not to confuse it with Pythium. Cernity and Strata Elite are the really strong products on it, but making sure you're not confusing those with Pythium. And that takes us on to Pythium. And the big takeaway of this that Dr. Deborah's talked on already is it's an MIC, not a fungi, which Henry loves it when I use an analogy, so I'm trying to wedge one in here, but essentially a flying fish and a bird both fly, but they're very different. And that's kind of what we're looking at with MIC, that they've just evolved from different pathways. So a lot of their underlying biological features are very different. They both infect plants and they both give similar symptoms, just like the flying fish and the bird are both flying, but they're actually very different underlying that. So probably the worst analogy I've used yet, so Henry will share to me in a minute, but you get the impression. It's going to affect all turfgrass species, and the big risk here is misidentification, misiding it with grey leaf spot. So the big takeaway would be, yeah, Pythium has that white cottony fungus. You just don't want to get it confused with grey leaf spot. And the reason for that is it's a very different biology. Heritage is our go-to, and the other products don't perform very well. So if you've definitely got Pythium and you want to be looking at those things that Dr. Deborah has talked about, you know, have you got very high levels of water? That's going to push that situation. Heritage would be the #1 go-to product, and the other products don't perform nearly so well because the biology is so different. And again, prevention is better than cure, so catching it before it gets out of hand. So yeah, much more simple with Pythium. It's about getting the ID right and then going straight for the heritage. So on to brown patch now. Yeah, so a few susceptible grasses, but including our perennial ryegrass. Again, recapping the top tips: air circulation and minimizing shade where that's possible, improving surface water drainage. There's been some evidence to suggest that pre-dawn irrigation that knocks down mycelia can show a reduction in brown patch severity, and where common, we would suggest using a fungicide to prevent brown patch because it can be quite devastating. And it does occur in all regions, especially where cool season grasses are grown, especially in transition areas. So obviously, traditionally, we're not considered a transition zone, but if in those stadium environments we keep seeing climatic drivers, we will artificially become a transition zone, because that's essentially where cool season grasses can reach the edge of their comfortable range, and so they're under a lot of stress, so it means diseases are able to come in. So that's one for the future potentially. Obviously, we're still a cool season zone, but you can see how we're going to be moving towards that transitional status in a sense. So this is a study done in the US. I couldn't get a study on ryegrass, unfortunately, but this one's on colonial bent, but hopefully will suggest similar results in a ryegrass situation. It was done in New Jersey by Rutgers University back in 2020. And you can see here strong pressure in the untreated control plots, so really high levels of disease pressure, up to sort of 70–80%. And in the applications they did with Acernity, really good levels of control. So we know it is a good product option for control of brown patch. So yeah, go-tos would be Acernity and Heritage, those systemic products that are going to go in, and the sort of middle of the road products, still good for intonation, would be your Stratolites and Medallions, but definitely leading with your Acernities and your Heritages for brown patch. We're here to help. There's a disease ID guide. We've talked a lot about, it's all about catching it early. So anything you can do to upskill your staff, you know, if it's just you or one other looking out for these diseases, you're obviously going to be more delayed than if you have the whole team looking for it. So you can get these disease ID guides off the website. They're really good. They've got a bit of information on each of them. Hopefully they're nice and user friendly. But the more people you can have out looking for these diseases, the earlier you're going to catch those symptoms and the more effective your treatments are going to be. And there's loads more disease information on the Syngenta Turf website as well, so there's more information on each of them and product recommendations as well. And as always, we're doing more work in this space. So we know it's a threat, we know it can be a very damaging disease if it gets out of hand, all of the ones that we've looked at. So we will be doing more work in that space, but do talk to us about things you're having problems with, and we can target our research in that way. Turf Advisor is a new free tool, which is going to be really powerful for those models. So that's something I just wanted to talk about. So in Turf Advisor, it's all free. You can just download it. It's got agronomic information for your site. One of the things you can do in there is tracking your GDD information. So you can just go into the GDD module for 0 degree and type in the date you want, and then it's going to tell you exactly how many GDD it's been since then, so you can figure out when you need to do your next premium application. So it's doing all the calculations for you. We also do have a grey leaf spot model. So far for the sites I monitor, I've only ever seen it be 0, because I've not been in a high pressure period, but essentially a model based purely on temperature. So it's not saying, if it says it's a 1 to 4 or 0 to 4 model, if it's not saying if it sees a 4, you will definitely see disease. I would take it to be, if you see a 4, you should be enhancing your level of scouting, because obviously you still need that bit of plant stress, you still need that leaf wetness. Temperature is just the biggest driver and the one that you can't control. So it's just helping you target when you should be scouting. And we know that leaf wetness and soil moisture are really important. And if we can get more data on these areas, we can build better and more powerful models to help us understand that. So if we can have data that's coming directly out of the turf in real time, then we can get much more powerful models to say, right, yeah, we think you're much more likely to see a disease outbreak at this time. Just touching on AIM, there's a new one-box fungicide solution that's available from this year. Really agronomically strong. We've got a lot of testing on this program, but we always see good results. It's included in the pack as a Cernity and Stratolite and Medallion, which can work together as an effective strategy. And there's extra turf rewards on that offer as well. So do speak to your rep about that deal at the moment. So yeah, we've covered loads and loads, and I can't recap it all, but definitely my big takeaway would be upskilling those who are scouting and getting any confirmation as soon as you can. So if you're not sure and you think, oh, I'm pretty sure that was grey leaf spot, it's probably worth getting it sent off and getting an ID done. And I know you can do that with Kate Entwistle, and you can do that with Deborah as well. But getting that confirmation can help you build that program that you know, right, it was grey leaf spot, so now we know what we need to do for next year. We need to have that on our agenda. Anything you can do to allow the leaf to dry out. You know, with COVID, we had these fire break lockdowns. If you can have time without moisture on the leaf, then that's going to be really powerful. It's going to stop that spread so rapidly. So any fire breaks you can put in your watering, as long as you keep plant health there, can be really beneficial. Increasing airflow, it's not an easy thing or an inexpensive thing, but some sites are considering looking at fans and anything to move the airflow. If you're looking for a stadium design in the next 3 to 5 years, it was something I would be considering as anything we can do to optimize airflow, because that is going to help. All of these diseases are made worse by the fact that there's no airflow or low airflow. A fungicide management plan, making sure what you do, reviewing it at the end of the year. Could it be better? Could it be tweaked? Could it be more preventative? Because we always see better results when we apply preventatively. And yeah, we want to do more leaf spot models. So if you want us to build a stadium model, then we need more data. We can't just do it on 3, because it might be that stadiums in Scotland aren't seeing 3 degrees, they're only seeing 1 degree increase. So we need to have that, and that might be something then we can build into Turf Advisor. So you'll be able to say, yep, I'm managing a stadium environment, you'll click that, and it'll always modify your whole data set to make it more relevant for you. So that's something we can do. We just need your data to help us build that. So that's everything from me, Henry.
Thank you, Sean. Very good. And no, I liked your analogy. I've got to say, for those of you who don't know, Sean, is actually his superpower is the ability to make up analogies, or you know, off the cuff. Usually they're inappropriate and off color. So obviously you tone that down for the purposes of this presentation, which I think we're all, you know, relieved about. But anyway, I've got a question for you. Which you probably won't know the answer to. It's one of these hospital pass questions, all right? So I'm just kind of putting that kind of thing in, but it's a good question. So the question is, because we talked a lot about sort of not overusing nitrogen, for instance, as a result of, you know, because it might inflame an outbreak depending on what disease it is. And the question is, moving on from that, what effect does Primo Max have, if any, have on the development of disease? You know, putting the sward into regulation. Do you know if that affects the progression of the disease or the performance of a fungicide applied to control the disease?
Yeah, it is a real hospital pass. You're true. Thank you for that. Well, we saw some interesting information out of Ireland, didn't we, earlier in the year, Henry, about Primo Max being a really good tool to keep the amount of nitrogen in the leaf higher. Yeah. So you get about a 1% increase from the data that they'd looked at in your leaf tissue nitrogen. So it's a really hard thing to get. You would have to apply quite a lot of nitrogen to be able to get the equivalent nitrogen increase that you get from using Primo Max. So they found that the tissue nitrogen level to be a really important indicator of how much disease you were going to experience. So I don't know specifically for the grey leaf spot. I don't know if anyone's looked at that, but for other turf diseases, it is actually going to help you because it's keeping more of that nitrogen you're putting on in the leaf, so your plant is healthier. And it all comes back to plant health, doesn't it? So if your plant is healthier, it's able to perform better. So it's a roundabout way of saying, I don't know for grey leaf spot, but on some turf diseases we see the plant health because you're spending less of that nitrogen, you're keeping it in the plant.
Fine, excellent, well played, sir. So yeah, I mean, like, a lot of this stuff, like, there's more that we don't know than we do know. So I think the inference was, could the use of Primo Max kind of adversely affect our disease control programs? And I think the sort of, the general consensus on that would be no. It's sort of like, obviously it's kind of like probably more important that it's in there than it's out, is what you were saying. Now, we had another sort of a specific question regarding to, not for you, Sean, but for me, actually, which was about the sort of, is ProSelect, our seed, resistant to grey leaf spot? And I think in terms of, I think because host is really important when it comes to any kind of disease control strategy. The susceptibility of your host is a massive point. Deborah sort of touched on the fact that sort of there's probably a lot of testing gone on in the States. There's maybe more testing being carried out in Europe now because it's more endemic there. It's easier to test. And I think testing will come out more and more and more. So I don't know particularly about the pluses and minuses of the ProSelect 1, for instance, but what we do in these situations is we recommend varieties that we really do know have good tolerance or reduced susceptibility to grey leaf spot. In our case, it would be that we supply Mistral, which I think is stellar in the States. And so we are conscious of kind of host susceptibility, especially kind of with the stadia environment being such a heightened environment. And I think it will just come into play more and more and more. It feels like we're at the early stages of grey leaf spot, our journey to walk through it all. And it is going to be an even more important question about host susceptibility in the grass type, the varietal kind of attributes. And so at the moment we go to specialist cultivars rather than it being sort of a key part of our sort of off-the-shelf mixes. Okay, if that helps. Now, is Deborah still on? Got one for Deborah, which I forgot to ask when I was too busy referring to my own notes. So this is quite a nice question, Deborah. Right, make sure you're on, your voice is on, because you mentioned at Brown Patch at the very start of your kind of presentation where we sort of, there's loads and loads of information kind of going on, but actually it was a point that sort of almost like we went past, but someone raised it. Because you talked about in Brown Patch, the mycelial bundles or sclerotia, was it, that sort of can, that can sort of act as storage or overwinter, can help sort of Brown Patch overwinter or be sort of, present during times when it sort of wouldn't be developing, and the question was, I get there in the end, how do we know if we've got this as a particular issue and what can we do about it if it is an issue, or is it just, I'll let you answer the question, Mark.
Okay, so my understanding is that rhizoctonias are fairly prevalent, so it's more likely than not that rhizoctonia species will be abundant in your root zone. Whether you have Rhizoctonia solani, which is the species which actually causes brown patch, that may be a little difficult to answer, but I don't know that there's actually great research summarizing in the UK, the species which are actually present, so it's a bit difficult to answer, but presuming that it is actually present, essentially when you core it off the top, you're going to remove a fair amount of potential inoculum, because essentially these sclerotia, they're on decaying tissues, so they fall off the leaves, and provided they're not punched deep into the profile, they're going to stay fair, I would have thought. Yeah. That they would stay fairly high in the profile. So yeah, at renovation, I think there's a good opportunity to remove a lot of that anyway. It's just when it comes to if you do have an outbreak and then you're overseeding, I think that for me is really where the risk would be a bit higher, I would have thought. And just to backtrack before I forget as well, someone asked a question about Primo and about plant growth regulators. I did actually have a comment on that. I think we need more data on that. And here's the reason why, the thinking behind why. If you have a weak pathogen in particular, the older most leaves are going to be more affected than the newer leaves. if you do not have new growth coming in to support the growth of that plant, it may suffer. So I think that we need a little bit more data. I'd be a bit hesitant in saying that it doesn't have an effect, but we need data to be sure of that.
Yes, well done. Yeah, the agronomist in me always kind of like, I'd be all right. Anyway, so good. The voice of reason comes in there, which is really good. Now, I just wanted to sort of get back to you on this actually, because the one thing that kind of did come out quite a lot in your presentation, Deborah, is about hygiene and about sort of, you know, simple practices of kind of like, you know, if you have an infected area to sort of, you know, to be careful about, you know, your movements through that area or from that area, and to kind of, you know, to have that, you know, to really take a sort of, you know, quite a careful approach to infections.
Yeah, I think it's just common sense, or at least it comes naturally to me. I mean, I'm a forensic scientist by trade, so my undergraduate degree, I learned this Lockhart principle, and it stayed with me for the better part of 20 years. And it really tells you that every contact leaves a trace, and it's something that I still use every day in my work. It's really, really important, because where you have mycelium, if you pick up pieces of that, you may carry it to another area. So it just makes sense, if you have a high risk area or an outbreak, simply approach it last if you can, if it's available to you. Maybe it's not, but if it is a decent strategy to at least limit the potential transfer. And just to, you know, if you're doing a stadium first with a piece of machinery and you have to take that same piece of machinery to a training ground, make sure it's clean. Don't assume that because it's yours and, you know, your great ground staff, it doesn't mean that you will not carry something from one location to the next, and vice versa as well.
Yeah, really good. Every contact leaves a trace. There you go. That's my take-home message. Anyway, that's good. So thank you so much. So look, that's it. I think we've cleared the questions. I think we are, well, we're 3 minutes late. Come on. I mean, that's really, really good. You've covered so much ground. You know, we've talked about the pathogen, we talked about the environmental factors, we've talked about identification and proactive monitoring, and then Sean has just finished up with prevention and control. And cultural management is an understanding of your of the full story is really what we're sort of talking about here, not just sort of simple kind of take-home messages or whatever. So there's a lot to unpack, there's a lot to think about, and you know, so we do need to keep talking with each other, we need to keep learning. Okay, so look, first of all, a big thank you to both our speakers. You did an absolutely brilliant job, both of you today, and I feel like I'm sort of certainly in a better place than I was at the start of this session. So thank you for that. And also, of course, thank you for everyone else for attending. You know, we really do hope that this information, you found it helpful, and that ultimately it kind of helps you in your day-to-day routines, you don't feel detached from what's being said, you know, that it's textbook stuff, that actually, you know, that you take home and you actually, you know, do start doing your proactive monitoring, and you do sort of like, you know, are more confident about identification and the ways in which you should sort of approach different stages of diseases and stuff like that. So look, we will, or the plan would be to share the presentations as much as we can. I am conscious that, you know, there are sort of some imagery and stuff like that that may be sort of like can't be forwarded, but we'll see what we can do on that, because I know that sort of like you're going to want to probably reflect on what's been said. The video of this, I'm sure, will be made available as well. It has been recorded, and although they haven't been supplied yet, there is basis points for those who need CPD points on this session. So look, we will be in touch about all those things to follow up from this session. But in the end, look, thanks to everyone for attending. I think it's been a great session, and we look forward to the next time. So goodbye all.
Thanks very much.
Bye, thanks everyone.
Yeah.